Now, when we look at the dominant and recessive relationships, we're going to have a look at an example of the round and the wrinkled pea, which are the ones that Mendel first worked with. Now, he always bred with what he called true breeding plants. Now, true breeding meant that there was no skipping of generations of hidden characteristics. You only got that one characteristic coming through. So if you take that into account when we think of how we apply that with two pieces of information, if each parent is giving a piece of information but every generation you're getting the same characteristic popping up, then it's safe to assume that the only piece of information that they get from each parent is exactly the same. Or, in other words, they're getting the same recipe from both parents. There are no hidden characteristics. So with the true breeding pea plant, if we assume that both parents have the same recipe, so with the round peas, and with the wrinkled peas, they both have wrinkled. Now notice I did not put a W for the wrinkled peas. That is because when you're dealing with dominant and recessive relationships in genetics, we always base the symbols on the strongest or most dominant trait that we're working with. In this case, round. So all of the symbols will be R's, but the dominant trait will be a big R for round, and the recessive trait will be a little r for wrinkled. We're going to now look at all the possible combinations that these traits could have mixed up in. Now, if each parent only gets to give one copy of a recipe to its offspring, this is the pattern that we'd find. Say that this recipe from this parent could combine with either that recipe or that recipe, which means you'll have the possible combinations of big R, little r in both combinations. Now let's look at the second one. Okay, now when you look at all of those possible combinations, each combination leads to a big R and a little r, which means even if the wrinkled pea trait is there, technically if it's the recessive one, then it shouldn't actually appear, which means you'll only see the dominant one coming through the plant, in other words, round. In other words, you get 100% big R, little r. Now, when you look at the data on this fits exactly with what Mendel had. His first generation of breeding round and wrinkleds was 100% round. Then what he did was he combined two of these F1 generations and interbred them to see what would come out. Okay, so when we cross a big R and a little r with another big R, little r parent, let's see what we get. We get one pure big R, two mixed big R, little r's, and one little r. Now, because big R is dominant, it's the only one that's actually going to show through. So even with the big R's and the little r's, the wrinkled characteristic here is hidden, which means all three of these are going to show up round. And this one here, the little r, is the only one that's going to show wrinkled. But again, it shows that that characteristic has skipped that generation because all of these were round. This actually shows that the combinations would end up exactly as Mendel's experiments showed, 75% round, 25% wrinkled. What it doesn't show you is that some of the round are what we call true breeding plants, which means they only have the big R, and some of the round ones are what we call, well, they're not true breeding plants, we actually call them heterozygous, meaning that they have two types of recipes for a specific characteristic. So here comes the terminology. Any true breeding plants, which means they have only one type of recipe, are called homozygous, meaning one type of recipe. Homozygous for the big R, and homozygous for the little r. In science now we say that they're homozygous for the round and homozygous for the wrinkled. When you have two different types of recipes in there, it's just that one is hidden, we call that heterozygous. In other words, you have both. Now the other thing that I will pull you into now is the names that we have for these. Now we've got here each one of these little recipes, so the wrinkled or the round, and a little bit more terminology. Each of these recipes for these characteristics that you carry, they are what we call genes. Genes are sections of your DNA that encode for a specific trait or characteristic. So you have specific sections set aside for every characteristic you have, whether it's eye color, hair color, blood type. So the actual recipe for a characteristic is called a gene, but as we know, each one of those recipes has many different versions of them. And those different versions, like round or wrinkled, are called alleles. So each version of a recipe is an allele. So you'll get used to using some of this terminology. We're going to go on now and have another look at an easier way we could have done this with what we call a Punnett square. Instead of drawing all those lines across the place to try and look at the combinations, the easier way is to split them up with what we call a Punnett square. And I'm going to do this example whilst using other terminology as well. So in our example we had a round crossed with a wrinkled. Now we would say that that was a homozygous round, so an R crossed with a homozygous wrinkled, because there's only one copy of each, or one type of each allele. These ones only have round, these ones only have wrinkled. So then we put this into what we call a Punnett square for each generation. So for the first one, so say this parent here, they could give either a big R on their allele or another big R allele. And this parent here could give either a little r or a little r. Now, then you can actually see what the combinations are going to be. If this parent here gives this one and this gives this one, your combination is going to be big R, little r. And again, like this. Just makes it a lot easier than drawing lines all over the place, and it means you don't miss anything. And as you can see, all the possible combinations are 100% heterozygous for round. Hetero meaning I have one dominant trait and one recessive hidden trait. So we call this heterozygous round. Over here we had homozygous round and homozygous wrinkled. And that was our F1 generation. Then we took the children and did them again so that they became parents. So say we cross this one with this one. Put one on the side, one on the top. This parent here could donate either a big R gene or a little r gene. This parent here could give a big R gene or a little r gene. Now look at the combination. Now this allows us to quickly get a picture of what it was like for the next generation. We have one homozygous round, one homozygous wrinkled, and two heterozygous round. Just like that. And these guys here would only show up as round. So that means our round was 75%, and these guys here showed up as wrinkled. Again, this demonstrates how Mendel's theory worked when you take into consideration that each parent can donate one out of two pieces of information to the child, and shows how traits can skip generations because they're hidden in a certain generation by other dominant characteristics. So these Punnett squares are an awful lot easier than squiggling it around with the lines like that, and they're a lot more clear too when you actually want to look at the percentages. So you'll be using Punnett squares to work out an awful lot of your genetic problems. Again, notice, can you find an allele? An allele is one of these little recipes, whether it round or wrinkled. The allele is the recipe, the version, and the characteristic, which is the gene, is for P shape, okay, and that's determined by a combination of whatever alleles you are given. So each one of these are positions on a gene. 2 genes with 2 versions of a recipe in this one individual. And this is really important in genetics, because if you have a recipe and it's corrupt for any reason, you're in a lot of trouble if you don't have a backup copy. So this constant, we have 2 copies of everything, one from each parent, means essentially that we have an extra chance if any one of our genes fails. And sometimes we do find that in genetics. Someone will actually have a recessive trait where they shouldn't, and often it's because the dominant trait, the recipe for that, its allele, has been corrupted in some way and is not working, and because of that, the recessive trait will actually take over and do the job instead. It's really important in an organism to have that backup. Okay, now we're going to use Punnett squares to work out incomplete dominance and codominance. Alright, well, you may not know it, but the budgie is a well-known pet in Australia, and people love budgies, and there are different colours. Main ones are blue, yellow and green. What you don't realise is that the blue budgie and the yellow budgie are the true breeding organisms, and the green budgie is actually an example of incomplete dominance, because as you know on your paint palette, blue plus yellow makes green. So blue budgies are what we call true breeding, and they have a blue allele from each parent. Yellow budgies are true breeding, and they have a yellow allele from each parent with their genes for colour. If you cross them though, you get a green budgie, and I'll show you how. So for your first generation, F1 generation, when you cross each one of these, each of them goes from homozygous to heterozygous. But heterozygous in this case does not mean that you have one allele dominating over another. Instead, your characteristic shows a new trait, in this case green. However, when we then breed those budgies from that F1 generation, we find that we get the old traits coming back if we breed them out. Now, I'm also going to show you here a new technique. We tend to get quite sick of writing alleles like this, and we tend to just denote them by a letter. The reason we have that line in front of us is to remind us that that's a section of a gene, a section of DNA. So as long as you get that in your head, you can actually skip drawing those little lines everywhere and just go straight to the letter. So as you can see here, we either get homozygous blue, homozygous yellow, or heterozygous, which turns out to be green. And again, then that allows us to easily figure out the percentages of each of these characteristics. And here we have homozygous blue, heterozygous green, homozygous yellow. Now I'm going to throw a new piece of information at you. When we look at what genes an organism has, we call that their genotype. When we look at what we actually see as a characteristic, because remember, you can't always tell what someone's genes are by the characteristic. In the case of the peas, they could be either homozygous round or heterozygous round. You didn't know what alleles they had to give that round characteristic. So we have a name for this. When you know their genes and you know what 2 recipes they have, what 2 alleles they have, we call that their genotype. When you're actually just saying what we see on the surface, we call it their phenotype, and we write that differently. Instead of BB, we write, what do we see? Blue. Here we see green, and here we see yellow, because this is what we actually see coming through. We call that the phenotypes. Unless you actually crossbreed across different generations, or look at the characteristics that have travelled from parent to child through at least 2 generations, you cannot figure out what genotypes are in that organism to show that phenotype. You need to actually look at it across generations in order to figure it out. To get away with flowers, I'm giving you an easy example of codominance in an animal. We often see roan cows around. That's these brown and white or black and white speckled animals. And what you don't actually realize is that they're an example of codominance. You breed roan by breeding a brown cow, true breeding brown cow, with a true breeding white cow, and you get a roan calf as a result. And here's the actual alleles that go with it. If it's a true breeding cow, it would have to have both brown alleles, and if it's true breeding white, it has to have white alleles. But notice neither of them are little lowercase, because both of these alleles are quite dominant and try to come through. When you put this into a Punnett square for the first generation, you realize the only combination you can actually make is brown and white together, which means you end up with 100% roan or BWs. So roan is essentially like incomplete dominance. They are heterozygous for brown and white alleles, whereas up here we had homozygous parents, and that's where you get the roan colour from. Homozygous parents breeding together to make heterozygous brown and white cows. Now, if you took two of these animals and bred them together, you would actually find a re-emergence of the original characteristic, and I'll show you how. Notice here, instead of just going brown cow and white cow with the alleles, I've just said I'm crossing a BW with a BW here. Now, when you cross these guys, you end up with either pure white, pure brown, or again the heterozygous version, which is roan. So when you breed that second generation, you can have 25% brown, 25% white, and 50% will still be roan cows. Now remember, here's the genotypes of the possible offspring, genotypes, and the phenotypes that we would see from those offspring would be brown, roan, and white. So you've also got to get your head around what the phenotype is, what we see, what the genotype is, what are the allele combinations that that organism has to give it that phenotype. In the cases of codominance and incomplete dominance, it's a bit easier once you know the pattern, because if you see a green budgie, you know it has to have a blue and a yellow gene. If you see a roan cow, you know it has to have a brown and a white gene in order to get that phenotype. So it's a little bit easier to figure out the genotypes, or the two alleles that an organism has in codominant and incomplete dominant relationships. It's not so easy to figure out the alleles in dominant recessive relationships. To do that, you have to do several generations of crossbreeding.